So first of all, I want to thank Carlo for the invitation to this workshop. I'm very amazed to be here. Thank you so much. Thank you for all the organizer. That's a very nice workshop you put together. And I know how hard it is to put it online. So congratulations for all of that. All right, so as Lorella said, I will be talking about looking for predictors of Rtms effect to enhance its efficacy. And I'm sorry, I will have to break the EEG party because I will be talking mainly about MRI. So we'll start with fMRI and then we'll move to structural controllability and then E-field modeling. So just to let you know, this talk has been done in collaboration between Duke and the NIH. So I'll present you work from both places. Oopsie, doesn't like it. There we go. So here are my outlines. So what we're gonna talk about is why do we need to enhance efficacy, right? And to be more specific, I will be talking mainly about online TMS. So when you do offline TMS, you will do a task and then you will do your TMS for several amount of time, and then you will do a task again. And just look at how your performance vary before and after TMS. This is kind of cool, but it's also just considering the cumulative effect of TMS. What I will be focusing on today is online TMS. So subjects are doing the task, and while they are doing the task, boom, we're gonna stimulate. We think that this allows a better precision in terms of understanding the timing of the cognitive processes. So I'll just be talking about online TMS because this is what we have been using in our studies, and so I'll just focus on that. So to understand why we need to enhance TMS, I will show you the result from a large meta-analysis we've performed on several cognitive domains. And then I will show you the predictors of our TMS efficacy. In a second step, I will present you new approaches that will allow to enhance our TMS efficacy. I'll be talking about network controllability, and then I will step aside a little bit and we'll talk about electric field modeling. So now, with no further ado, let's jump into this talk and let's see why do we need to enhance TMS efficacy. So this meta-analysis, I've been doing it with my colleague at the NIH, Dr. Zi Deng. And so we looked for TMS effect on cognition. So for the keywords, we were only interested by online, by online TMS, as I said. So we look for online TMS, but also concurrent or even related TMS, because it's what people tend to refer to for online TMS. And of course, TMS was either TMS, RTMS, repetitive transcranial magnetic stimulation, all the options. For the cognitive domains, we used very large words, so cognitive tasks, cognitive processes, cognition, but then we also search for very specific tasks, so memory, working memory, attention, etc., etc. So we looked in several databases and we ended up with more than 9,000 citations. And trust me, that's a lot of paper to go through. So to be more specific in our search, of course, we use inclusion and exclusion criteria. And what I will be focusing on is just that we looked for paper only working with healthy controls. We didn't include any patients because that could be a confounding factor. We look for online RTMS, no surprise. I think I repeated that to you several times now already. But we only included study using figure of 8 coil TMS because we know how different the electric field is when you use, for example, a round coil. For the comparators, we look for studies that compare active TMS to sham TMS. So sham TMS, we know that's a big word that can encompass a lot of different approaches. So we included everything: an active control site, an electrical sham, a study you flip the coil, study you put another coil below the coil. But what we didn't include is all the study that didn't use any stimulation, like no stimulation as a comparison. In terms of the outcomes, we looked at the reaction time and the accuracy on the cognitive tasks. For the timing, of course, we looked at the tests performed during our TMS session, and we included all types of study design except study cases. So after our full text screening, we ended with 126 articles, which is more handleable than 9,000. And so what did we find? Here are the results. So the first table is just the accuracy. It's presenting you the effect size of active RTMS over sham TMS across all the cognitive domains that we found or defined, such as attention, executive control, imagery, language, memory, motor task, or perception. And this is showing you the TMS, different TMS frequencies. So 1 Hertz, which is known to be inhibitory, and then 5, 10, and 20 Hertz. What I'm showing you here, I'll just move that away, sorry. Okay. So that's the effect size. So everything in blue indicate that active Rtms disrupted performance compared to sham stimulation. So as you can see on the accuracy, TMS mainly disrupted performance, and that's the same effect on reaction time. But the main point I want to highlight here is our dark gray cells. So what you can see here is all the study using TMS and showing no significant difference between active and sham TMS. And as you can see, there is a lot of dark gray cells, right? There is about 50%. So 50% of the study cannot find any superiority of active TMS over sham stimulation. And that's why we need to enhance it, right? We need to find better parameters. So as we all know, when we use TMS, so many things we can do. When we stimulate, we can stimulate at intensity based upon motor threshold, either active resting, phosphine threshold, and we can also use the maximum stimulator output, so just a fixed intensity. And just as a reminder of yesterday workshop, Dr. Zolturi showed us that about, I think, 90% of the study use intensity based on motor threshold. And we replicate this here because only 27% of the study are using stimulator output. In terms of stimulation frequency, we can use 1, 5, 10, and 20 Hertz. We need to know which one to use according to the cognitive task we are using. As you can see here, 1 Hertz Rtms is underrepresented. And this is because it's very hard to fit 1 Hertz, this slow rhythm, when you're doing a cognitive task. And there's a lot of variability in all the other ones. Then you need to know where you need to target. So often this will be guided by what you are doing. So let's say I'm doing a visual task, of course, I will focus on the occipital cortex. However, if I'm doing a memory task, then I will be stimulating the frontal cortex. And of course, in TMS, everybody loves DLPFC. So that's why DLPFC is kind of well represented in all this study. But once you define your target, you need to know how you will target it. And so first thing, you can use a neuronavigation system or not. And we can see that the majority of study use it, which is great. That's a big progress in the field. But we need to know how we want to target these areas. So the most common or older, I would say, is to use scalp measurements. So that would be, for example, the 5 centimeter rule for depression studies. And we can also use some hotspots. So again, if I want to stimulate the visual cortex, then I might want to do some phosphine thresholds. That will be hotspot stimulation. I can also use anatomical MRI, which is more individualized, or fMRI. And so what we all know, I think, since 2009, since CIMeC and collaborator published this paper, is that fMRI targeting largely improve Rtms efficacy. So on this paper, they directly compared the effect of 4 targeting approaches. So the first one was P4, so that's just the 10-20 methods. They also use Taderac group coordinates, anatomical MRI, or fMRI. And what they found is that only when they use fMRI targeting, they found significant improvement with active Rtms compared to sham stimulation. Even more interesting, they compare the number of subjects that would be needed to reach a significant threshold. So here, this p-value, this magic p-value, right, of 0.05. And so they found that when you're using the 10-20 methods, you need about 45 subjects to reach a significant threshold. However, more sophisticated are your targeting approach and lower this number of subjects is. So for example, when you use fMRI, you just need 5 subjects to reach a significant threshold. So that's interesting. That's definitely super cool. We love fMRI targeting. But on the other side, this study was just based on 5 subjects per group, so it's slightly underpowered. And so what we did with our meta-analysis was just looking at that in a way larger sample size, right? And we indeed found that targeting approach predicted the Rtms efficacy. So here I just plotted the effect size. So of course, 0 means no effect, and the lower you go, the better the TMS effect is. And what you can see is that when study using hotspot stimulation, we're the one leading to the lowest TMS efficacy. And then these get better with more sophisticated approach until reaching the fMRI targeting with the strongest effect size. So how to enhance TMS efficacy? Well, let's use fMRI. That's kind of pretty straightforward, right? But we can wonder if this is enough, right? Because when you think about fMRI targeting, you will think about the subject performing a task, maybe activating a specific region of his brain, and you'll be like, okay, I need to stimulate this area. But is it really the case in the brain? I don't think so, right? The brain is an ensemble of brain region that communicates together, that forms network, that also interact within network, and it's not just one single region. Martha this morning showed this beautiful slide that I copy paste, sorry about that, but I found it very pretty, which found that indeed TMS effects tend to spread out to other brain regions, and that spread out more and more with time. What I want to show you as well is the result from this review paper that we published in Neuroimage pretty recently. And those are the RTMS effect on resting state connectivity. And so what I'm showing you here is all the brain networks, so somatomotor, ventral, dorsal attention, limbic, frontal limbic network, etc. And what I want to show you here is, so the arrow will be where you send the pulse, and the end of the arrow will be where the effects are. Oopsie. And so what you see here, for example, when you stimulate the somatomotor cortex network, you induce changes in this network. That's good, that's what you want, but that's not it. You also induce changes in the ventral attention network, the dorsal attention network, the default mode, all the other networks. So yes, TMS effects are focal. It's good to use one spot to define what we want to target, right? But since it's spread out so much, is there any measure that we can use that take into account this communication within the brain? And so this measure is structural controllability. All right, so now let's move to our next step, the new approaches to enhance our TMS efficacy. And let's start by talking about structural targeting. So controllability, wow, that's a weird term. What is it? Well, you have to know that it's pretty common in the engineering field. Everybody uses that. And this is more recent in neuroscience, right? People started to use it like a few years ago. And now what is it? It's using these white matter bundles. So you will just acquire a diffusion weighted imaging, and then you will parcellate your brain in several brain regions. In our cases, we used 471 regions, but we can use more, we can use less, it just depends on the atlas you're using. And then you will make a matrix counting the number of streamlines connecting each pair of regions. This matrix A, which is right here, structural controllability said that you can move the state of the brain from this current state to the next one if you input some energy, some signal into your system. And what matters here is where you input the energy and which type of node you are stimulating. So if you input your signal onto a node which is called an average controller, then you will move your brain into very easy to reach state. And this is confirmed by this result from Gu and collaborators, who found that the average controller were located mainly in the default mode network. However, if you're stimulating a node that is a model controller, you're moving the brain into very hard to reach state. And once again, this is confirmed by the location of this model controller. So for example, here you can find them in the frontal and in the parietal area, which are the executive areas of, I'll make it better, brain areas of executive functions. All right. So I showed you that controllability is related to difficulty. Now, is it true? And can we confirm that with TMS data? Okay, so what I showed you before was all about review paper, looking into the literature and so on, but we also did some work. And so I'm gonna show you our empirical data to answer this question. So this is our task. So we use online RTMS to enhance working memory performance. We might think we are kind of crazy because we all this disruptive effect that we found before, that's kind of not straightforward. But because of our previous study from Dr. Luber, Dr. Lysianne, we trusted in this ability. So what is this task? The subject will see this array of letters. So for example, DTRP, and then these letters will disappear and we'll have a delay period of 5 seconds. During these 5 seconds, we ask our subject to alphabetize the letters. So for example, with DTRP, they will now be doing DPRT and repeating that and keeping them in working memory. And then they will see a probe, which is the letter with a number above it. And the question is, does the number above the letter correctly represent the position of the letter in the new alphabetized order. So for example, here in the DPRT, P was indeed the second letters. So subject will just say, yes, it was. For the TMS, we used, no surprise, individualized fMRI targeting. And so we tried to target the DL, well, we didn't try, we did it. We targeted the DLPFC. This is just the group map activation. So as you can see, there are different spots. And of course, when we do that at the individual level, the spots can be different between subjects. For the TMS part, we use 5 Hz Rtms because we know that when you're performing a working memory task, the theta band communication is very important between the frontal and the parietal areas. So 5 Hz Rtms, we stimulated at motor threshold during 5 seconds, either before the encoding to kind of help the neuron prepping for the task, or during the delay period when the subject are actually manipulating information. What did we, no, not yet. To make the task even harder, because that was already complicated, we also added 3 difficulty levels. So in this task, and as you can see on this slide, difficulty level will just be the number of letters on the screen. And since it's individualized, some subject will have, let's say, 4 letters at the easy difficulty level, while higher performer will have 5, 6, or 7 letters. And then we'll just increase by 1 letter in the medium and by 1 more in the hard difficulty level. So this task is very complicated. What did we find in terms of RTMS effect? This is showing you the accuracy with active RTMS in dark gray and sham TMS in light gray. And what we found is an interaction between stimulation, so active or sham, and difficulty level, easy, medium, and hard. The decomposition of this interaction showed that we didn't find any difference between active and sham on the easy difficulty level. We didn't find any difference on the medium difficulty level, but we found a significant improvement with active RTMS over sham stimulation only in the hard difficulty level. So that's kind of cool because we want to play with model controllability, which is related to difficulty. So now we have a TMS effect only in the difficult condition. So what we can wonder is, does this RTMS effect is predicted by controllability? If this is true, that model controllability indeed help pushing the brain in how to reach state, then the individual with highest model controllability should benefit the most from RTMS. So what we did is just computed this matrix, so RTMS effect, which is just the accuracy with active RTMS minus sham RTMS divided by sham RTMS. So it's just a normalization. And just to show you on this graph, 0 will mean that there is no difference between active and sham TMS. Everything above 0 means that active RTMS enhance performance compared to sham stimulation, and inversely for below 0 data. And what we found is a significant correlation between RTMS effect and model controllability, as we expected. And what you can see is that the subject benefiting the most from RTMS are the ones showing the strongest model controllability values. So we demonstrated that controllability indeed is indeed correlated with TMS effect. And which is even more interesting is that this correlation is present only for the hardest condition. We didn't find it for the easy difficulty level or for the medium difficulty level. And just FYI, side note, we didn't find a correlation with the degree, which is another graph theory measure, which indicate that really it's specific to controllability. Now, if you will, let's just go back on our fMRI activation. So what we did to target is to use a parametric increase associated with difficulty. So we used all these difficulty levels and just created our parametric bold signal. And once again, if controllability is indeed related to difficulty, then we should expect a correlation between both. Is it the case? Yes, it is. We have a significant correlation here again, proving that once again, yes, model controllability is related to difficulty. So I showed you that controllability is related to difficulty, it correlates well with TMS, and it works on the DLPFC. What we can wonder next is, what about the site and the task specificity? Is it really specific to these two regions, to this one region? Can we make it better? And so to answer that question, I'll show you our second study. So we use the same task for the first one, which is our alphabetization task. And we use a second task during which subjects do not have to alphabetize, they just have to maintain the serial position of the letter. So for example, if I show them DTRP, they will have to rehearse that, maintain that, so DTRP, DTRP, DTRP. And when the probe shows up, they just have to say, was P the fourth letter in this serial order? So this task is way easier to do. And while the DLPFC is highly involved in the alphabetization process, we believe that the parietal cortex is more involved in the maintenance process. So we stimulated these two areas, both on the left side. And then, spoiler, by the way, no TMS effect on that. Active and sham TMS didn't differ. At least it was not significant. But we still looked at the correlation between our TMS effect and model controllability. So what I'm showing you here on the left part is everything about the dorsolateral prefrontal cortex for the alphabetization task at the top and for the maintenance task at the bottom. The thing on the right will be the same, but on the parietal cortex, alphabetization on the top and maintenance on the bottom. What we expect to find, if what we found earlier was true, is to replicate this effect. So we expect to have a correlation between our TMS effect and model controllability in the alphabetization. But because the DLPFC is less involved in the maintenance process, we don't expect any correlation here. And this is what we found. Okay, it's not significant, I'll give you that, but it's close. And at least it's heading in the same direction, right? So that seems to be pretty interesting. Reversely, with the parietal cortex, as I said earlier, we expect it to be more involved in the maintenance process. So we expect our correlation to be right here. And this is the case. Very close to significance, 0.05. We are getting there. But what we found is that controllability is very specific to where you stimulate, what you are doing, and this is a double dissociation between site and task. But so now, so all of that was about healthy young adults performing a task, which was hard, but doing okay for them. What we can wonder is how to reach state important for depression? Those people, they really need to get better with TMS. They really need TMS to work for them. They really need to reach those how to reach, AKA feeling better states. Does it work for them? So we know that when you use TMS to treat depression, we have several options. The first one, and most common, I think still, is just the 5 centimeter rule, which was improved by the 5.5 and on the 6 centimeter. But it's also associated with often missing the DLPFC as proven by some papers. Option 2 is to target with the 10-20 method, so stimulating F3. And again, it's better because at least you take into account the difference between head size, but it also misses a lot of information about brain physiology and anatomy. Third option is the resting state connectivity. So we have this work from Mike Fox, Sean Siddiqui and all in the Boston's lab. And this kind of cool, I really love this job. And what they found is that when you stimulate the DLPFC at an area which is strongly connected with the subgenual anterior cingulate cortex, then the TMS effects are stronger. This is just a post hoc analysis. So they just stimulated with BM3 and they ended up calculating the connectivity between those two areas and they found a significant correlation. So that's cool, that's an option. But when we do resting state, there are also so many parameters to control, right? Do subjects need to keep their eyes open? Do they need to close them? Do they need to be watching at a cross? Are they thinking about something that might disrupt? I think, can you still hear me? My earphones. Hello?
Yes, we can hear you.
Okay. Okay, they just did a weird noise, sorry. So yeah, resting state is cool, but it's not that easy to apply. And so what we want to say is, can we use controllability? Can controllability predict the TMS effect on depressive symptom changes? This is just a post hoc analysis, but what we found, we had 25 patients with major depressive disorders. We stimulated with the approved FDA protocol, so 10 Hertz, 120% resting motor threshold. And the DLPFC was stimulated according to the BEAM F3 method. And what we found is, again, a significant correlation between the changes in depressive symptoms and model controllability. And once again, individuals benefiting the most from RTMS, so improving their symptoms, are the ones showing the highest model controllability value. So that seems to work. I showed you that controllability is predicted by TMS effect on the DLPFC, and it's really specific to difficulty. I also show you a double dissociation between site and task, and I show you that it also works with depression. So what we can think now is that maybe you can just place your subject into a scanner, collect your DTI. That's pretty quick. That's pretty straightforward. They don't have to do anything. They don't have to worry about anything. Collect your data, find your node that has the highest model controllability value and stimulate it. That's something we need to try. But for now, I will pause on the targeting and I will step aside and talk about the electric field modeling. All right, so when I talk about electric field modeling, probably the majority of you are thinking in terms of, oh yeah, coil orientation. We want the current to flow in this specific direction. We want the current to reach and hit the sulcus perpendicularly to it to get the stronger TMS effect. Electric field modeling is mainly used for that. But what I want to talk about today is, can we use electric field modeling to define stimulation intensity and not location? Why? Why would we need to do that? Well, because a lot of study, as I showed you, about 90% of the study tend to use intensity based upon motor threshold. That's cool, that's easy. You will find your hotspot, look at your motor threshold and stimulate either above or below this intensity. But when you're doing that, you are not taking into account the differences in head anatomy and brain physiology between different brain regions, right? I think the motor cortex is very different from other brain regions, for example, as the DLPFC. The motor cortex is hyper excitable compared to it. It will just respond faster, respond stronger. The DLPFC is kind of, slower behind it. So probably we are, I think, understimulating when we are using resting motor threshold. So now that we saw the why, how do we do that? Well, the answer is just about computer simulations. We talked about it yesterday a lot by using CIMeC, for example. And so I will show you here the approach, like what we can do. I cannot show you a comparison of intensity based upon resting motor threshold, intensity based upon E-field, because we didn't do it yet. I will just show you what we did with E-field and if it works or not. So to do that, we used data from our previous study in which we stimulated, as I said, at 5 Hertz at resting motor threshold, the DLPFC. But in this study, we wanted to work on the parietal cortex. So we first defined parietal mask that was based upon fMRI activation. And then for each subject, we calculated the E-field magnitude for each voxel within this mask. We then ranked them by their E-field magnitude. As you can see here, that's just an example from one subject. So E-field going from 0 to its maximum value, and then the number of voxels showing this E-field magnitude. And we then look for the 100th voxel with the largest E-field magnitude, which in that case, I cannot see because I have the stuff above it, but you can see the value on your own. I think it's 55 volts per meter. And then we did the same stuff for 9 subjects that we had at this time. So that's just for those 9 subjects. And we computed the average of this E100 matrix, which ended up being 56 volts per meter. We call this number or E-ref or E-field of reference. So now we knew that we needed to stimulate the subject in our new study at 56 volts per meter, or at least at a stimulation intensity that will reach 56 volts per meter in our parietal target. Okay, and so what? Well, we use computer simulations again to transform this information into DI/dt, so current information for each individual subject. And then to make it even fancier, we also adjusted for hair thickness. So we know how much distance matters with TMS, right? They tend to dissipate with distance. So it's important, I think, to take into account this value as well. So we use this gauge to measure the hair thickness compression. And then we computed DI/dt for different value. So let's assume in this case, the hair thickness was 1 millimeter. Then we knew that we needed to stimulate such as the current was 73.6 ampere per microseconds. And so then we transform the DI/dt information in maximum stimulator output, which is a more common value. And to do that, it was very basic for us because the device we are using are reporting the intensity in MSO in DI/dt. So we just had to do like a little scatter plots to know the transformation factor. And then what we did is compare two stimulation intensities. So either 100% EREF, such as the current was reaching 56 volts per meter in the brain, or 80% of that, so lower intensity. So these are our results. So again, we used the same alphabetization task. We stimulated the parietal cortex. What you see here is the coil location for each individual subject, and the arrow are representing the coil orientation. So I didn't talk about it because that's not the goal of this talk today, but we also use E-field, of course, to define the coil orientation. What we found is, once again, an effect of TMS only in the hardest difficulty level. But this time, instead of improving performance, active RTMS actually disrupted performance, moving from 57% accuracy to 55% accuracy with active TMS. Okay, that's the overall, that's the main effect results. Now, how did it work with the intensity? So what I will be showing you is, once again, our RTMS effect here. So once again, the 0 means no difference between active and sham. Everything below 0 will be the performance disruption with active RTMS. And as I told you before, we found disruption. So we expect our subject to be on this below 0 part of the graph. I'm showing you two intensity that we compare, the one at 80% of ERF and the one at 100% of ERF, reaching 56 volts per meter. And what I'm gonna show you on this graph is the RTMS effect for each individual subject. So here, what we can see, so we had 15 subjects in the study. We can see that on the red line, that's the mean, so the average across all the subjects. And as you can see, it's very close to 0. The standard error encompasses the 0, so that's not significant. And we had 7 subjects that has performance impaired after active RTMS compared to sham TMS. Now, what happened with 100% ERF? Well, that looks better, right? The mean is different from 0. It's also almost different from 80% ERF. And what we found is that more subjects are impaired by active RTMS compared to sham stimulation. Interestingly, when converting again the intensity as a percentage of the resting motor threshold, you can see that the majority of the subject received subthreshold stimulation. And I think this is kind of coherent with what Zolturi presented to you yesterday, showing that lower stimulation intensity are enough to induce significant changes in the brain. So the take-home message from this slide is, well, you can also use electric field-based modeling to define stimulation intensity. It's new, so we don't know exactly which value we can use, but it seems to be working. And so future directions, let's reconcile these two parts of the talk that I presented to you today. I showed you that our TMS effect is specific to the task condition, especially the difficulty of the task you're using, fMRI activation, electric field value, network controllability. And the future direction will be, can we use electric field modeling to assess the value in a white matter bundle? And can we combine that with network information, since we know that TMS effects spread out to the whole brain like that? So we need a lot of smart engineers in there to help us with that. But I think this is where the field is going. So to summarize, I show you why we need to enhance TMS efficacy. Well, because it's highly variable. That's the topic of this workshop. And because of the limited effect size of TMS. For the how to enhance it, I show you that fMRI targeting is an important factor and probably will help enhancing efficacy. And for the new approaches, what I showed you is that structural controllability is probably a very important factor. And I think we might want to go ahead and start using it as a way to target for TMS. And finally, I showed you that electric field modeling can be used not only for the coil orientation, but also to define the stimulation intensity. So now I would like to thank my team. So I was at Duke before, so Dr. Applebaum, Dr. Simon Davis, who helped me a lot with this talk, so thank you for him. My colleague, Li Fu Deng, Courtney Crowell, the E-field team, Moritz Danauer, Engel Peterchef, Wesley, and all our awesome coordinators. And I also want to thank the NIH with Dr. Luber, Dr. Lysianne, and Dr. Deng. And I'm so glad to be working with them now. So thank you everybody for your attention, and I'm happy to take any question if you have some.